You can hear me okay? Yeah, so not only biomaterials within the LRSM has seen enormous growth, but around the country, including NSF, one of the newest divisions within DMR is biomaterials. And in fact, they've been around for just a few years, and we're trying just now to formulate a philosophy and opinion for years going forward. So I'm going to give you our view, at least a few labs that I was asked to represent in actually 10 minutes, even though it's also a 30-minute talk squeezed into 10, right? So LRSM, when I came here, sitting down with Mike, think different. Yeah, don't think like everyone else. And so it's titled, the title of the talk is Biomaterials, but in fact what you'll see is more about materials approach to biology that comes back to biomaterials. And the argument is that we have to understand biological systems better from a materials perspective and use tools like AFM and X-ray diffraction. A number of tools aren't commonly used in the med school, right? To better understand biology in order to make better materials that might translate. Right? Motivation might be, well, biology has been around a few years longer than the LRSM, maybe more than 50 years. So let me from that perspective have remind you a bit of history of polymer science, right? So polymer science started in biology too, right? Isolation of polymers like polyisoprene or cellulose, right? Biochemical molecules that were then, as Wikipedia, right, the fount of all knowledge now, right, says that it became semi-synthetic with modifications of natural polymers leading to, eventually totally synthetic polymers, but starting with biochemistry, modified, right? And 90 years of such work before it was actually understood by Staudinger, a chemist, what we were really dealing with biochemistry, which was polymers, right? What we now know as molecules that are monomer by monomer assembled, right? And then 90 years later again with things like Nobel Prizes that we mentioned before. So as I said, I'm going to give you a bit of a very quick materials view of biology, and we'll see in some ways how this begins to come back into biology, come back into biomedicine. So here's a polymer science view of tissues, okay? That's really emerged just over the last decade with Paul Janmi and myself, I think in some ways leading the effort. So adult solid tissues are soft. Poke your skin, poke your various tissues. If you could poke your brain, whether you're human, mouse or rat, brain is always soft. It's always extremely soft. When I say that, you'd struggle to make a hydrogel as soft as brain. It's less than a kilopascal, all right? And you want to know what a kilopascal is in terms of soft matter physics. It's 0.001 kBt per nanometer, the molecular scale. So that's a very soft material. And as you go across the different soft different tissues, even cartilage, which you might think is stiff, at least at the micro scale, we're talking about materials from that elasticity perspective that are kilopascal, dozens of kilopascal. And then you ask, well, what's establishing this? And the most abundant polymer in you and I is collagen, all right, in animals in general. And collagen scales from brain through muscle and these other, as we'd expect with the elasticity. And in fact, it's quadratic. You can even recapitulate this in a dish, although the prefactor is wrong. Is different from what you get in tissue. This for example, is an organ in you and I. It's a heart. It's actually a chick heart, but you can take a detergent, get rid of all of the cells, and it still looks like, well, a ghost of a heart, right? That's the collagen you're looking at. In fact, that's 90% collagen from mass spectrometry, among the other physical tools that we commonly employ in our biological studies. So these are soft microenvironments, right? Measured in kBt per nanometer cube that cells adhere within. Doesn't matter. Why am I telling you this, right? So let's bring this understanding, which is surprisingly new, to biology, and that's how do cells respond. Right? And this is a field that's just emerged in the last dozen years. And so I'll give you a couple slides of examples. So here, you take a polymer gel, you make a semi-synthetic, like the whole field of polymer science started. You interpenetrate collagen 1, the most abundant polymer in you and I, into this cross-linked polymer gel. We control the elasticity classically by controlling cross-linking, right? So you culture cells on this, and Paul worked on this, started working on this about a decade ago, and shows that neuronal branching, neurons growing in a dish, branch much more, this is 5 or 6 times more, on a soft substrate like soft matrix like that of brain tissue than it does on a stiff gel or rigid glass. It adheres in all cases, but you get better neuronal physiology when you mimic the softness of brain tissue. We had shown with muscle a couple of years later that the striation, the characteristic assembly of the cytoskeleton, was optimal when you match the stiffness, the softness, stiffness, the intermediate stiffness of muscle, as opposed to too soft or too stiff. Same materials, different cross-link. And then others have picked up on this. This is a group up at Harvard, uses completely different gels and different ligands for cells to attach to. And they looked at bone development and came to the same sort of conclusion that a soft material that promotes bone growth that we know from biology is best for what you do in a dish. Companies sell lots of dishes of polystyrene that are very rigid. But this tells you do better biology if you pay attention to the mechanics of the material, of the tissue. Right? So we a few years ago asked the question: could you get these kind of behaviors with cells that can become these different cell types? A stem cell, which has a lot of interest in med schools around the world. And so here we took an adult stem cell that adheres, and people over the years had reported the ability to generate, to some extent, neuronal processes or muscle-like states or even bone states. And so this is that elasticity scale. And this adult stem cell, indeed, over hours and days, turns into the different lineages to an extent, not completely, based on the ability of the cell to generate forces and feel the elasticity. Right? It's recognizing as a deaf and blind person might, right, in a tactile way, what its microenvironment is, and starts turning on the proteins and the genes. Right? So we've gotten more and more into the biology here. And we find opportunities all along the way to make new kinds of measurements, from mass spectrometry I've told you about, to AFM, the mechanics. And here we're looking at using remarkable commercial arrays where you're able to lay out 1 million spots, this sort of density, 5-micron spots, to interrogate the whole genome. But we had found that no one had done simple things we learned from surface science decades ago, show that you get saturation-type binding and work out the signal-to-noise. And so that's what I show here, and we're the first person to do this, first group to do this kind of analysis. And you can get higher signal-to-noise and show that at the gene expression level, neurogenesis, these are a bunch of neuronal genes, myogenesis, muscle genes, osteogenes are being turned on by matrix elasticity in this very specific way. So you're rewiring the whole cell according to what it's adhering to at the gene expression level, right? And you can do this better and better now. So how is this occurring? So those are observations, right? So I'll give you a little bit of sense, and in the spirit of the LRSM, we always look to a theory and simulation. Well, we often do when we can, right? So we had observed, Paul had observed with his neurons, we had observed with these stem cells that the cytoskeleton, the structure within, right, the cell is strongly modulated by the softness, stiffness of that microenvironment. And so here one sees more diffuse and progressively more organized from stiff to rigid glass cytoskeleton, right? And so over the years, we've talked to people like Tom Lubensky, colleagues at Weizmann, Sam Safran, who all have this strong background in soft matter. How might we explain this? So liquid crystal elastomer ideas, active liquid crystal ideas have emerged, and things like, most recently, explanation of the striation I told you about in muscle as an active smectic phase, right? Maybe motivating, 50 years now, not dead smectics, but live smectics, right, that can self-assemble in response to things. All right. So I'm running out of time. I can tell you this relates to nuclear structures, but I've really run out of time. I just want to quickly go through, because I promised to touch on Les Dutton's work. As old as the LRSM is, lipid membranes, lyotropic, we heard a little bit about it earlier in the morning, is also similarly young field, or old field, right? 50 years old that we have this bilayer model. And we worked on these polymersomes and done a lot of simulations with Mike Klein over the years. And I'm going to skip the measurements and the simulations, as well as work on phase segregation, where we try to mimic the segregation because of natural membranes with heterogeneity and metal ions, both at the experimental level with a number of people in physics and chemistry and engineering, physiology, Paul Giambi included, and simulation with Mike Klein, and try to just skip through all this and tell you that Les Dutton also works in this biomimetic. He's part of his lab, a major part of his lab, studies membrane proteins relevant to bioenergetics, and then says, how can we understand this in a sense, but also engineer and simplify? And so, in fact, my wife works in that lab and provided this slide. And so the complexity that's here is simplified down to self-assembling peptides with things like porphyrins that insert in very engineered ways, right? And so over the years, they had a number of achievements, several reincarnations of the IRG, that you could functionally assemble these, insert these into membranes onto various surfaces as well, these mimics of these very complicated proteins, and have all sorts of functional readouts for gas sensing and electron transfer. All right. So I don't have lack of time to tell you about further biomedical applications like shrinking tumors and such. But my own interaction with the LRSM has been about 15 years. I've been here just past 15 years. And it's allowed us to take this materials biology approach to biomaterials, which is a bit different from most approaches. You see and you make. No. We see, we measure by novel means, and then we try to make something, right, as simple as possible. Right? So it's allowed us to take in biology seriously as a science, into the materials with materials and methods. To this day, you won't find an AFM in our med schools, as big as it is, right? They're all here, and they're in many labs. And also, wherever possible, in the spirit of the LRSM, bring in theory and simulation. And so I just expressed my gratitude, actually, to the founders and all the directors that kept it going along the way, as well as colleagues I've worked with. Okay, so that's my 12 minutes.